Well, Dan, thank you so much for joining me today. It's a bit of a snowy day here in Toronto, and I really appreciate the time you take.
No worries. Thank you for inviting me.
No problem. So, as I was we were just talking just before we started that, we met at A4M, and this has become, you gave a talk at A4M on this topic of plasminogens. Is that how it pronounce it? Plasmologens or
Plasmallogens.
Yeah. Plasmologens. Okay. It seems like I was talking with somebody else. This has become like just came out of kind of nowhere into the medical community, at least into the consciousness of the medical community. I thought, oh, this is very interesting because it's pretty much a breakthrough kind of technology. But maybe before we even get into that, if you could just give the viewers a little bit of understanding of who you are and what's your background, and
sure.
That'd be great.
So my background, my undergraduate studies is in synthetic organic chemistry. So I'm an organic chemist from the core, and then went into biochemistry, looking at the neurochemistry, the biochemical mechanisms of neurological disease. So my PhD is actually in psychiatric medicine from University of Alberta, there in Canada, the Neurochemical Research Institute there in Edmonton. And so from that basic understanding of the biochemistry of the brain and understanding how psychiatric diseases affect, neurological function and the neurological biochemistry, I went into work in the pharmaceutical industry, looking at mechanisms of actions of drugs. We're looking at neuroactive steroids, the GABA receptor modulation. And basically in the late 80s, 90s, when the genomics revolution was really getting into the big swing of things, my background was in biochemistry, looking at the small molecule aspect of life. And there wasn't really any counterpart to this genomics revolution where we could sequence the entire genomes of species and look at how one gene function works versus another gene function. And that's when my first main invention was a technology called non-targeted metabolic profiling. And this used a high field mass spectrometry, used an ion cyclotron to use ultra high resolution, ultra high mass accuracy, such that we could resolve basically all of the small molecules in a biological sample. And then we could do this in a non-targeted way. And from that, we used it for functional genomics research, a lot of basic research. But when we started, so we can measure literally thousands and thousands of small molecules in any kind of biological matrix. And then when we started applying that to human health disorders, and over a huge swath of programs, discovered the biochemical basis of many diseases and biochemical prodromes. Basically, we originally thought that, I originally thought that it'd be a very valuable diagnostic tool to be able to understand biochemically what is going on in a given disease. And that's actually quite valid. So the technology is very, very powerful. We use it still today in my labs here in California now. But it is, the original thought was the disease is causing the biochemical disturbance. Okay, so you have Alzheimer's disease, and the Alzheimer's disease is creating a biochemical disturbance. And this was the original thought, saying, okay, obviously, if I can see person A with healthy and person B that's not healthy, we live in a we live in a universe of biochemistry. So if I can visually see a difference in phenotype between individual A and individual B, there must be a biochemical correlate of that difference that we see. And that turns out to be true. Okay, people with colon cancer have a different biochemical profile than those without. People with children with autism, multiple sclerosis, different psychiatric disease, we can differentiate from bipolar, schizophrenia, and unipolar depression states, Parkinson's, Alzheimer's, and so on. So that's all actually true. But what was really the groundbreaking moment actually happened in colon cancer, not in Alzheimer's disease. Alzheimer's is another program with psychiatric issues. We did a study in Japan looking at the biochemical profile of people with colon cancer versus not, thinking the hypothesis was that after we surgically remove the colon tumor and the people recover, then the biomarkers should recover.
Come back to normal.
Right. Well, that didn't happen. What happened was the biomarkers stayed exactly the same after surgical removal of a tumor. And we said, well, this can't believe this. So we redid the entire study over again in a completely different universities, set of collaborators in Chiba University in Japan, a few hundred miles away, got exactly the same results. And this was the first real indication that these biochemical changes were not being caused by disease, but they pre-existed the disease, and they were creating a biochemical predisposition or a prodrome. So we had this temporal relationship where biochemical changes were occurring, and these biochemical changes then created the increased risk probability. And that really came down to the bald tire hypothesis, basically. It's like a bald tire it's not a guarantee of a flat tire, but it increases your probability of one. And so using that kind of technology, so when we went to Alzheimer's disease, okay, the biomarkers that were showing up as depleted in Alzheimer's were these molecules called plasmalogens. These are phospholipids that are critical for neurological membranes and all the membranes of the human body. And this followed a similar line of thought in that this was correlated with the severity of cognitive impairment. And
what are these serum plasmalogens?
These are all in the blood, yeah. But we've measured in the brain. Plasmalogens have been known for a long time. And we've known about them regarding they're obligate to human life. Like you must have plasmalogens in your membranes or you will not survive. Or if you do, you get born with dwarfism. Like there's many diseases of paroxysmal impairment in children, Zellweger's, Refsum's, rhizomelic chondrodysplasia punctata, many diseases, and these children do not survive very long because of the severe deficit of not just plasmalogens, but other paroxysmal related metabolites. What really wasn't of understanding was what was happening, fast forward 50, 60 years from birth, and these plasmalogens were starting to be depleted or they were going down in individuals. And this depletion of plasmalogens were correlated with other disorders. Alzheimer's, cognitive impairment is a big one. They're also, low levels are also associated with higher risk of Parkinson's. Virtually all cancers have low blood plasmalogen levels, and for different reason. So this is where the plasmalogen hypothesis really took off, and understanding the biochemistry of this membrane structure and these critical molecules that no one had heard of, even though we discovered them over 100 years ago, and we've been studying them extensively in different areas for a long time. So they're not brand new, and they're not trace. 20 to 30% of your entire brain lipid volume are these plasmalogens. And so essentially the brain shrinkage, the brain dehydration, when the brain turns into a raisin from a grape, that dehydration is really a delipidation of the brain. Okay. And so what happens to the brain is it loses its lipid volume. And one of the core lipids of the brain are these plasmalogens. And then when you lose that core morphological structure, you start losing function. And then again, you create the stressor on the system, and you're going to see slightly different manifestations in different individuals based upon their other underlying genetic predispositions to one disorder or another. So anyway, so that's where the plasmalogens come in. And then from that aspect, so I was first to discover that these low plasmalogens were occurring later in life, and that these low plasmalogens were linked to a series of diseases. The canary in the coal mine is Alzheimer's, or loss of cognitive impairment. And that's specifically related to the cholinergic system in the brain, which is exquisitely sensitive to low plasmalogens. And coincidentally, it's also the key neurotransmitter of your neuromuscular junction, which is why sarcopenia, or muscle wasting, and cognitive impairment, or frailty, if you will, go hand in hand. And so maintaining plasmalogen levels are also critical for maintaining muscle mass, neuromuscular performance outside the brain. So the cholinergic
You're saying like acetylcholine is related to these plasmalogens?
Oh yeah. So the maintenance of it in the brain are dependent upon plasmalogens. Right. So and it has to do with the cholinergic chemistry of the neurons. And so when you look at the brain, people think of the brain as a computer, but it's not a computer at all. It's more like the wiring harness of your car, where you have a pipe or a conduit with many, many wires in it. And these wires are connecting, neuron A to neuron B. And this wiring harness, in your car, for example, there might be one wire that goes from your taillights to your brake pedal, right? But in your brain, and it goes across that through the course of your car. But in your brain, that's called your corpus callosum. That's your big pipe of neurons that go from one area, one hemisphere to the other. And that has 200 million neurons in it. So 200 million wires are going through there. And so you don't have just one wire from your taillight to your brake pedal. You have probably 100 or 1,000 in your brain.